Hello class, this unit's about cells. There are 7 standards and also 7 videos. So I broke them up. Instead of teaching it as one long, crazy, extended video, I thought maybe I could split up into 7 videos that are about 1 minute to 5 minutes. It'd be easier for you to comprehend. So this first standard is you understand what do they use to study cells. If you know that cells are really tiny, you know that they require more than the human eye. Specifically, what they use are microscopes, and the microscopes they use can get us nice pictures like this of cells. Normally, if you look at this chart right here, from 10 meters to about 100 micrometers is where we could see what we can see. Anything smaller than 100 micrometers is where we stop seeing stuff. At that point, we use something called light microscopes or light microscopy. And anything lower than that will need either super resolution or electron microscope. So really this whole standard is, do you know the difference? Do you know what is a light microscope and an electron microscope? To help you with the difference between the two of them, there's a couple of tables I have for you guys. This is what electron microscopes, the pictures that electron microscopes show. So you can sort of see like the detail, the picture, etc. What you probably have been used to if you use microscopes like at school or in the labs, is you see pictures like this. They're flat, they're static, you can see the outside of the cells and things like that, but they're not as detailed as an electron microscope. So some comparisons, how light microscope works, if you've used a regular microscope at schools, like there's a light that shows from the bottom and it passes through the specimen. On the other side, an electron, what they do is they focus a beam of electrons and it shoots back so you can see what is coming back. So one has light going through it and one has a beam of electrons that go through or onto a specimen. The light, because it's refracted, is magnified in a light one. And it magnifies up to 1,000, whereas electron can magnify up to 1,000,000. In light, it could be alive or moving. For an electron, it has to be non-living or in a vacuum. Light, you can do color, and electron, you primarily only get black and white. The two types of electron microscopes, there's transmission electrons and then there's scanning electrons. So they call them TEM and SEM. So in electron microscopes, there's two branches. Transmission is 2D. It creates a flat image with a lot of detail, and they use like staining atoms or heavy metal dyes. So this is an example of an image from a TEM or a transmission electron microscope. Scanning is for detailed studies, and it gives great field of depth. So think of it as like the awesome digital camera. Like a cell phone camera versus like a nice digital camera. This is a scanning electron microscope. So this is not a flat image, but actually, looks almost 3D. So for studying structure and function, this is the last part of this section, is we could study it in 2 different ways. One's called cell fractionation. Just like it sounds like, fractionate means we're going to take apart these cells and then we're going to separate the major organelles. So we're going to talk about organelles. Organelles is a word for tiny organs. Okay. Another way is called ultracentrifuge, where they apply a force 1,000,000 times the force of gravity to separate the cell organelles with the most dense at the bottom. So here's a ultracentrifuge process, how it looks like. So if we took essentially, if we follow this timeline here, if we have tissue cells, like if we took muscle tissue or anything, homogenization, which means homogenize being the same, they sort of make it in a way, this looks like a blender, so that we have a consistent, this mixture is pretty much the same. Here's that centrifugation. They spin it to apply a force that's 1,000,000 times gravity, and you can see over many times, so this is 10 minutes, 20 minutes, 60 minutes, and 3 hours, you'll see different parts of the organs of the cells. So to study a cell, you use a microscope, but then you could also use cell fractionation or ultracentrifuge, or centrifugation, to actually see the parts of a cell. So that's standard 1. Hopefully it was very simple for you. You should be able to understand the tools and the strategies that they use to study the cell. Hope you enjoy.